were less likely to have used up all the iron. But we will have more confirmation of this when we get our results from the iron concentration measurements. And how else can we find out the differences in community structure? So the phytoplankton produce different pigments based on the species. So we are going to be measuring those pigments, species-specific pigment, using HPLC. We are also going to do genetic analysis on our samples, and because certain species of iron are more tolerant, certain species of phytoplankton, are more tolerant to low iron concentrations due to their genetic adaptations. And it's been shown that iron addition can change the gene expressions even on a very short time scale. So our future work will be to investigate those differences through the genetic analysis and see exactly how the phytoplankton at the two different stations change. So, in summary, the importance of light and iron for phytoplankton differs depending on the community. At 46 South, light limitation affected the phytoplankton more than iron limitation. And at 65 South, iron limitation affected the phytoplankton more than light. And understanding these differences can help us model the effects of climate change better. Thank you very much for your attention, and thank you to APECS for organizing this conference.
Okay, thank you very much for that talk. Again, in the interest of time, unfortunately we don't have time for questions just now. We're going to move on to the next presenter, and we're going to stay in the Southern Ocean. And Erica Carley is going to talk about insights into trophic ecology of the Antarctic silverfish in Antarctic coastal ecosystems.
Okay. Can you see?
Yes.
Okay. Ma si fa togliere? Sorry, one moment.
No problem.
Okay. Okay. My study is insight into trophic ecological of Antarctic silverfish, program Antarctica in Antarctic coastal ecosystem. Sorry. Okay. Program Antarctica belongs to the suborder Notothenidae, which is the most abundant of Antarctic fish, and currently includes a single family, Notothenidae, according to the last Scientists' Committee on Research Antarctic, and comprising 135 species, including 106 Antarctic and 29 North Arctic. This fish has adaptation, has presence of antifreeze glycoproteins, low quantity of hemoglobin and red blood cells. Like all Notothenidae, lack swim bladder, large amount of lipids, and reduced skeletal ossification. Among Notothenidae, program Antarctica is the only known holopelagic species. Program Antarctica is a circumpolar pelagic Notothenidae fish, which is very abundant in the coastal water of Antarctica. Live soul stage or flight in the water column at depth from 0 to 900 meters. It's dominant in the water of continental shelf, were represent 98% by the number of the total ictioplankton on the Weddell Sea continental shelf, and 90% of the pelagic fish biomass, and 98% of the fish larvae in the Ross Sea. The life cycle of the Antarctic silverfish is characterized by ontogenetic shift and in vertical and spatial distribution. Adults are mostly distributed in mid-level water, while eggs and young larva stage are an important of archaeopelagic community. Actually, in the Ross Sea is the only nursery area described. The evidence of direct interaction between the early life stage of Antarctic silverfish and the sea ice cover is highly relevant in terms of implication for prediction of the possible impact of environmental change on silverfish population and the coastal Antarctic ecosystem, where this fish plays a central role. It is the prevalent plankton feeder of the intermediate trophic level, and it plays a pivotal role in the trophic web, being the link between the plankton and the community of top predator. Sampling were caught during 4 surveys conducted within international collaboration projects in different sectors of the Southern Ocean. 83 fishes were collected in the Ross Sea in 2004 and 2008 during New Zealand survey. 62 fish were collected in the Dumont-d'Urville Sea in 2008 near the French station Dumont-d'Urville, and 23 fish were collected in the Weddell Sea in 2012 during the German survey. From the analysis of the stomach content, resulted that in the Ross Sea, the diet program Antarctica is composed by a great number of prey species. As we can see, the most common prey are the copepods, Medrida gerlachei, and Parocheta species. After, there are other copepods and other planktonic invertebrates of different taxa. A similar composition of prey is found in the stomach of sample caught in the Dumont-d'Urville Sea. Instead, in the stomachs of the sample for Weddell Sea, I found a few prey species, mainly the euphasids, Euphausia superbia and Euphausia cristallorhophias. To highlight difference in diet co-composition, I have grouped the various prey in 3 representative groups: copepods, euphasids, and other prey. In the Ross Sea and Dumont-d'Urville Sea, copepods are the most common prey, while in Weddell Sea is an overwhelming dominance of euphasids.